All right. So hello and welcome. I am Hayden Fennell. I'm this year's coordinator and organizer of the 2024 um, CompuCell Virtual Tissue Modeling Workshop and Hackathon. Today's orientation session will be focused mainly on giving you some background on course logistics, a few things you may want to do to prepare for the workshop, and then we'll do a quick demonstration walkthrough of CompuCell. Some notes up front. This slide deck is available in the student materials folder at the following link. I will put these in the chat for you here. But again, if you need anything from the slide deck today, you can find the slides here. This is the folder that we'll be using to send out all of the materials for the duration of the workshop. Right now, it's mostly just orientation information and some reference materials, but you will see that folder begin to populate over the next week as we start to upload our slide decks for the workshop. Um, please submit all of your questions and concerns today through the Zoom chat. I'll be monitoring it periodically, and there'll be a couple of spots where we can stop, give people a chance to ask questions if they have them along the way. We do have a Slack space for the workshop this year. I'll go ahead and put that link in the chat also. We do recommend that you join that in advance of the workshop. We'll have a slide coming up later about what we use the Slack for, but it's a good place to get questions answered and talk to other participants. So we do recommend that you join the Slack. Last note here, workshop sessions throughout the workshop will be live streamed, recorded, and distributed on YouTube. The current session is not being live streamed, but it is being recorded, just so everybody is aware of that. This session will be uploaded to YouTube later today and then sent out by email, so people who can't make it or couldn't make it will have a chance to see the recording. That's true of all the workshop sessions as well. All workshop sessions will be available after the workshop for future viewing. Last important note, we are not offering a remote CompuCell option this year like we have in previous years. We will just be using local installations of CompuCell for the workshop. So it is really important that you get CompuCell downloaded and installed on your personal computer before the start of the workshop. There will not be much time during the workshop to help troubleshoot installations. So if you are having difficulty with getting CompuCell installed properly, or if your installation is not working for some reason, please do let us know as soon as possible so we can help you get that sorted out. If you don't have CompuCell installed yet today, don't worry about it at this point. There will be an example later that you can just sort of follow along with, but there will be instructions on doing it yourself afterwards if you'd like to try it on your own as well. So with that, again, just a quick overview. Today we're going to talk about course logistics, some different types of background knowledge and reading you may want to do kind of in this week before the workshop if you have some time. Things to do to prepare for the course, hackathon logistics, and then, like I said, we'll do a walkthrough of building a simple model in CompuCell. This is a quick note. So this number is actually incorrect now. We have over 220 people registered this year. So we will do our best to support everyone, but we are a relatively small team. So please do be a little bit patient with us. We generally have a policy of responding to emails both before and after the hackathon within 24 hours. But we do tend to get a lot of emails, especially in that week leading up and during the workshop. So if for some reason you haven't heard back within 24 hours, please don't hesitate to just send your email again. Don't feel like you're bugging us. We try to do our best of staying on top of all the emails, but sometimes things do get lost or things get grouped together or sent to spam sometimes even. So please do feel free to reach out to us again if you haven't heard back again within 24 hours. So if you're attending the workshop again, or if you already have some background in CompuCell and just start attending to learn a little bit more, get some more experience. If you're interested in helping to sort of TA with some of the sessions, we'd be happy to bring you on board. So please just let me know at the email address here. I'll put it in the chat as well. If you are interested in actually helping out with any of the sessions. So with that, welcome. This is sort of the first official meeting, I guess, of the CompuCell workshop. Thank you everybody for being here today, and we will try to get through this in a relatively timely manner. So we just want to start out with a couple of notes on kind of what CompuCell is and what it does. So what we'll learn to model over the course of the workshop. CompuCell is kind of focused on doing sort of two things in particular. It's been used to do a lot of different things, but there's kind of two areas that people use CompuCell for most heavily. The first one is developmental biology simulations. So this is basically the question of how do we go from a fertilized egg and then basically to an adult organism and the steps in between. So CompuCell is often used to simulate developmental processes, and it's often used to simulate study of how different conditions affect disease prognosis and the mechanisms behind that. It looks at things like developmental diseases, infectious diseases. We can use it to study toxicology. So how do certain compounds cause damage? Why do they cause the damage they do? And the larger goal of this is sort of this idea of bringing some predictive power into medicine and bioengineering. So how can we control, repair, create new forms of the processes once we understand the mechanisms behind them, and how do we target medicine and treatment towards helping to alleviate or prevent those issues. One of the big motivations behind this, especially right now, is that this offers an alternative to animal testing. Not necessarily in all cases, but in a lot of cases, we can use computational simulations to replace some of the need for in vivo studies. And it also allows for easier tuning and manipulation of variables to test a number of different kind of parameter sets and how it affects the time evolution of different biological processes. Excuse me. So just to walk through, well, first, to give an idea of the kind of the scale of simulations that CompuCell is targeted at. So CompuCell is a multi-scale modeling software. If we look at kind of the broad scale of biological simulations, starting at the largest scale, we would consider kind of the whole animal simulation, sort of a digital twin of an entire organism. Next level down would be the organs that make up those organisms, which are then broken down into various types of tissue, which can be further broken down into, groups of cellular aggregates within the tissue. We can start to go even lower than that. So you have intercellular signaling between cells. Can be another level of scale. You can look at individual cell behavior given different simulation setups, and then you can even look at subcellular behavior of different components within the cell. So CompuCell, if you see the square of the dotted line here, CompuCell is sort of situated within that tissue level style of analysis and simulation. It can be used to do some intercellular signaling. There's an add-on package, which we'll talk about during the workshop, that allows you to do network modeling within CompuCell. But for the most part, CompuCell is kind of situated at the size of the tissue and the organ. Just to give you kind of an idea of where CompuCell is most useful. And with that in mind, we can walk through a couple of examples of things that have been done in the past with CompuCell. So this is sort of just a broad overview of the various types of things you can do with the software package. It's been used to study polycystic kidney disease in the past. So this is looking at cyst development on the renal tubules in the liver. I think this is in response to acetaminophen poisoning. Actually, so you can look at how you go from kind of normal renal tubule to cyst development over the course of time. It's also been used to look at tumor interaction with blood vessels. So how, as the tumor is growing within vasculature, how it kind of disrupts the vasculature and how it saps nutrients from it as well. Here you can see in red, we have our vascular system, and in green, we have the tumor growing within the vasculature network. And these are all from published papers. You can see the citations here if you'd like to read more about them. Slightly different studies, it's been used in diabetic retinopathy therapy optimization. So if we look at where to target the laser treatment during diabetic retinopathy therapy and different patterns of laser treatments, how that affects the prognosis of tissue healing in different ways. And it's also been used to study retinal, sort of again with the retinal theme, retinal neovascularization. This is where blood vessels actually start to grow into the back of the retina, can cause retinal detachments. This is a pretty serious issue. You can see here in the video, the red tissue represents the vasculature growing into the retina. The green is the retinal tissue. And you can see on the side here where the retinal detachment occurs and then actually causes a tear over time. You'll see it start to form here. So a few other examples of things that have been done outside of our lab. We have a computational model predicting disruption of blood vessel development. So you can see here over the course of certain parameters, blood vessels start to fall apart and deaggregate. Models of computational modeling and simulation of genital tubercle development and how different initial conditions affect the progression of the development over time, as well as the computational model of secondary palate fusion and disruption. So this is looking at the formation of palate tissue through development and kind of what causes disruptions of that, which lead ultimately to the disorder known as cleft palate. So if the palate is disrupted during formation, how that results in cleft palate, and if we can understand that mechanistically, we can target treatments towards that more specifically. CompuCell has also been used to study viral infection and immune response. During COVID, there were a number of studies using CompuCell to look at how virus spreads throughout various types of tissue. I won't go into too much detail about that today. This is just to give you an idea again of the various settings where CompuCell has been used in the past. So lots of different types of applications, all using the same underlying simulation methodology. So to get into kind of the learning objectives of this course, so by the end of this course, you should be able to look at a biological problem and ask quantitative questions that can be answered with simulation. So you should be able to form useful hypotheses that are actually addressable using simulations more generally, but CompuCell3D in particular. You should be able to analyze the components of a multicellular system as well as interactions and then translate them into executable CompuCell code. And then you should be able to build and modify CompuCell simulations as well as display and analyze the simulation data to derive biologically meaningful results. So by the end of the course, you should be able to engage with more or less the complete modeling process, going from starting with a biological problem that you're interested in studying, developing questions, turning that into a model that can be studied, potentially developing or finding some math that helps model that, and then turning that into computational simulation that you can use to answer those initial questions or hypotheses. Particularly if you participate in the hackathon will be a good opportunity to practice working through this entire process. All right, any questions before we get into course logistics? So one question in the chat. CompuCell only support agent-based modeling, or does it support other tissue mechanics modeling like hyperelasticity? So I'm not super familiar with the principles behind hyperelasticity, but I would say that these two things are actually not mutually exclusive. Agent-based modeling is more of a modeling approach. Hyperelasticity, I think, would be a property of the tissue themselves. We can talk maybe more specifically at the end of the orientation today, if you'll be around at the end, Abhishek, about specifically what you want to do with CompuCell. But there's a number of different things we can do with how to set up your cells to behave the way you want them to. Any other questions before we move into course logistics? How would you relate the ROS by Open Robotics and CC3D software? So that I am not sure about. Karthik, we can talk more about that in detail later. We may um, have a session or two on developing CompuCell extensions. Um, that's the only way I can think to do that because I'm not familiar with the ROS stuff. What does ROS stand for? Um, yeah. Okay. So yeah, we'll circle back on that later. That may be something we have to talk about by email later, because it does sound interesting. Okay. So I think you can go ahead and continue to leave questions in the chat. We'll come back around um, later. Is there a chance to simulate a metabolic model? Yes, you can definitely do that in CompuCell. We'll talk more about that. All right. So just to go over some of the course logistics, what the workshop will actually look like. So again, the workshop will be completely virtual this year. It'll be all by Zoom. We have links coming for that soon. There will be a link for the workshop and a link for the hackathon. We will send those out um, by next week at the latest, but they will also go up on the workshop website at least one day before the workshop starts. So if you lose your email or can't find it or something like that, or for some reason you can't access the link that we send, it will be posted on the website as well for backup. Excuse me, sorry. I meant to mute, but it didn't take. That's awful. Okay, my apologies. So there will also be a hackathon teaming document that we'll be sending out tomorrow, in addition to a hackathon interest survey. So we'll be polling your interests um, on what you may or may not want to participate in for the hackathon. That'll go out tomorrow. So keep an eye out on your emails tomorrow afternoon for that. And there's also a hackathon channel in the Slack workspace where you can start doing some teaming in advance of the workshop if you'd like. Just a couple notes on Zoom logistics. I know everybody at this point is probably pretty well versed in using Zoom. So I'll try not to be too detailed about that, but there are some notes going in. Sorry, scratchy throat today for some reason. But so screen sharing and microphones will be disabled by default when you join the meeting, just because we have so many people attending this year. I want to avoid any confusion or complexity with microphone muting, but you should have the ability to unmute if you did want to ask a question. We do ask, though, that again, because there will be so many people in each session, that you put questions in the chat first. For each session, there will be an instructor, a moderator, and a TA. The moderator and the TA will be monitoring the chat for questions. Some questions can generally be answered just through the chat. But if it's something that's especially relevant to what the instructor is discussing, or if it's something that we feel the instructor should address, we will forward it. We'll break in and ask the instructor. And then at that point, if we need clarification, you can unmute and ask your question that way. If there are technical problems with Zoom, please let us know right away. We want to make sure that everybody has their Zoom client working. One thing to note is that it's important to make sure that your Zoom client is not only working properly, but is updated to the latest version. Typically, we have issues if people's clients are out of sync with the meeting host, their version number. Some features get disabled, like polls, certain kinds of reaction emojis. So being able to raise your hand for some reason gets disabled if your client is out of date. So there's some things that get disabled if your client is out of date from the meeting hosts. So we recommend just trying to update your Zoom client a day or two before the meeting. Zoom does have a tendency to push out major updates kind of unexpectedly sometimes. So if we're made aware of a big update, we'll make sure to let people know to update their client in the evening after the workshop has ended for the day. But just do try to make sure that your client is up to date so that all the features work, because we will be using those polls to gauge completion during activities. You will need access to those polls throughout the course of the workshop. Exercises that we do during the workshop will be made available in the Google Drive so that you can continue to work on them offline if you'd like, or after the workshop, if you'd like to revisit them later. Videos and PowerPoints will also be made available in case you miss any materials. And like we said, the sessions will be recorded and posted to YouTube. So the live streams typically take an hour or two to process into a permanent video. But usually within an hour or so, the live streams will be available. If you weren't able to watch them live, you can watch them within an hour or so after they've processed into a normal video. And we'll send out the links for those each night. The links aren't generated until the video is processed into a permanent video. So we'll send those links out each evening for you to look at. But we'll also give you a link of where to check during the day in case you're trying to watch asynchronously for where each live stream is. Just a quick overview of some of our people. So like I said, I'm Hayden Fennell, the coordinator and organizer. Professor James Glazier is also the co-lead of the workshop. Our primary teachers this year will be Gilberto Thomas, Pedro Sensi del Castel, Professor Julio Belmonte. We also have Dr. Jim Sluka, Priya Madhyapuk, and TJ Sago doing a number of sessions this year. And Jim Sluka and TJ Sago will also be available periodically to help with technical issues throughout the week. As far as who to contact goes, if you're not sure, if you're having an issue or a question, you're not sure who to send it to, you can always just send it to me first. And if I can't answer the question, I will forward you to the appropriate party. So when in doubt, just email me. And we will either work it out together or figure out who we need to talk to get things solved for you. So daily schedule. This is just kind of an overview of what each day of the workshop will look like. These times are all in Eastern Daylight Time, which is UTC -4. This is the same time zone as New York City. So if you're not sure how to translate that into the current time, just translate your time into New York City time and you will be on the right schedule. We'll start the meeting each day. We'll open the meeting at around 9:30 a.m. We'll have 15 minutes where we kind of designated official discussion and question time. So if you have something that you'd like to bring up with the instructors or coordinators before we start for the day, there will be some time to do that. Like I said, we officially start that at 9:45, but we usually open the meeting at around 9:30. So if you've got a question that you think may take a few minutes to answer, you can join anytime after 9:30 to ask that. We'll start our official module sequence at 10 a.m. each day. Module 1 will go from 10 a.m. to 11:30, so each module is 90 minutes long. About half of that will be introduction to the necessary content you need, and then another half of that will usually be hands-on exercises, working with CompuCell code, different types of activities. After that, we'll have a 15-minute break, move into Module 2 from 11:45 to 1:15 p.m. After that, we'll have a sort of extended lunch break. We do from 1:15 to 2 p.m. We have an extra session where we have 45 minutes. Sometimes we use it for hackathon teaming exercises, doing presentations. We have a couple of different activities planned for that extra session this year. And then from 2 to 2:45, we'll have sort of an official lunch break. Everybody will have a chance to go eat. So the idea here is that if you want to go get your lunch to eat during the extra session, that's fine. But there will be some time to eat during lunch break. I know for the people in our time zone, 2 o'clock is a little bit late for a traditional lunch break. But we found that this is the best time that works given how many different time zones people are coming from um, for that lunch. Then we'll resume module 3 for the day at 2:45 to 4:15, another 15-minute break, followed by the last module of the day from 4:30 to 6 p.m. And then we'll have 30 minutes of open question time for people to ask questions either about the content that we discussed that day, anything from previous days, if you have questions about upcoming content, or if you're having issues with something, troubleshooting some code, or if your installation broke or something like that. This is just open time for you to come and ask all those questions. Sometimes we can stick around a little bit longer if we need to. But generally, we do try to like to conclude by around 6:30 to give our instructors some time to prepare for the next day. So we know that these are long days. So if you do need to go take care of other things, or if you have to miss a session here or there, that's okay. Again, the recordings will be available after the fact. We do recommend that you try to make as much as possible of the first 2 days of the workshop, because that's where we'll kind of cover a lot of the basics of how to actually use CompuCell. But again, if you do need to, attend to other things throughout the workshop, we understand that with the time zone differences and with people's summer schedules, things get kind of complicated. So we do offer the sessions asynchronously if you need to catch up yourself later. If you're having issues with some of the content, if you are trying to catch up asynchronously later, please feel free to email us. We'll try to get you some resources where we can to kind of help you keep up to date if you did need to miss a day or something like that.
Applications what?
All right. So any questions about the course schedule before we move on? We'll talk a little bit about the hackathon in the second half of the orientation. Okay, so feel free to continue to put questions in the chat as we move forward. I know sometimes it takes a minute to type everything out. I'll take a look at that later. All right. So if there's no questions on the actual schedule, we'll get a little bit into some areas where you may want to do a little bit of background reading if you have some time this week before the workshop. So there's kind of, for computational modeling in general, but specifically with CompuCell, there's kind of 5 areas that we think are important to have a little bit of surface level knowledge. So you don't need to know all these topics in depth, but it is helpful to know a little bit about them in advance of the workshop. Again, just so that if you're familiar with the vocabulary and some of the jargon and things like that, it makes learning some of this content, especially in a fast-paced course or a setting like a workshop, makes it a little bit easier, we've found. So there's kind of 5 areas: general principles of modeling, some Python programming. You will definitely need a little bit of Python to work effectively with CompuCell. Obviously, with computational biology, you want to know a little bit about some of the biology involved, some of the biophysics involved in the modeling of that biology, and then the mathematics, obviously, that help with that modeling. So these slides have a number of things on them. Don't panic. We will cover everything that you need to know during the workshop. The purpose of these slides is just to give you some starting points of what to maybe start thinking about in advance of the workshop if you did want to do some background reading to do a little bit of advanced prep. So these prompts are, it's not, not that you need to know all of these things in detail. They're kind of, again, just points to get you thinking about things that if you've never really thought about this before, maybe some things to start looking up. So for the background principles of modeling, probably the most important point here is this idea of defining questions that can be answered by models. So how to come up with questions that you can actually address with the modeling software, because not all questions are modelable. We'll talk a little bit more about that during the workshop, about how to come up with those questions. But again, conceptualizing and constructing models, a big part of this is deciding what components and mechanisms are important to your model. All models are abstractions to some degree. There are pieces of the real world phenomenon that are not included in the model. and deciding which of those components are necessary and which of them are not is kind of an important skill to start thinking about at least before um, really getting into the modeling process. Understanding the limitations of models is useful as well as how to do a little bit of basic model validation and falsification. So how to determine if the model is telling you what it expected it would tell you, but then also how to determine if that value is, physically accurate and correct. These are kind of two different pieces of the validation question. A little bit on the background of Python. So this is the piece that will probably be the most important to participating in the workshop as effectively as possible, is understanding a little bit of Python coming in. And again, we offer our Python bootcamp. That'll be on July 28th this year. So if you have never done Python before, or if it's been a while since you've done Python, we do highly recommend that you try to attend the bootcamp. If you can't attend the boot camp this year because it's on a Sunday, we know that sometimes people have conflicting obligations. We do have last year's boot camp recordings available. That'll be sent out tonight. The link to that will be sent out in an announcement email later today. So if you would like to watch last year's boot camp in advance of the workshop, if you can't attend this year's boot camp, we recommend doing that if you're not already familiar with Python. But some things you need to know, basic syntax and indentation. How indentation works in Python is pretty important. So understanding that, the basics of loops, particularly for loops, and along with that, Boolean logic and conditionals, how to do if statements, how to check things within a loop. You know a little bit about object types, how objects and inheritance works in Python, the basics of lists and dictionaries, basics of what a class is in Python, particularly the self-dot class. CompuCell uses the self-dot function from Python pretty extensively to keep track of its cell list during a simulation. So knowing how self-dot works is useful. And then also a little bit about attributes and methods. So again, we go over all of the things you need to know in our Python bootcamp. So if any of these things are looking really unfamiliar, then we recommend that you do come to the bootcamp. Again, if you can't, check last year's videos. And if you're really just not sure what to do because you can't do either of those things, send us an email and we'll try to get you some resources. Again, you don't need to know a ton of Python, but there are a few key things that you'll need to know to write CompuCell code effectively. So a little bit about biological background. Again, you don't need to know all these things in detail. These are just some prompts to get you thinking before the workshop. So obviously, CompuCell is a multicellular modeling software, so it's good to know what are cells, what do they do, how do cells generate forces, i.e. how do they move around, how are tissues constructed, so how do cells aggregate together into useful arrangements. Just some other things, what's the extracellular matrix, how does it interact with cells, how do networks relate to cell behaviors, so chemical signaling networks. And kind of the last point here, how do biologists represent information? So what do diagrams that normally come in biology papers and textbooks, sorry. What do these sort of cartoon diagrams, as they're called, actually represent? That will be useful, especially if you're planning to participate in the hackathon. Looking through literature is kind of a part of that. So getting a feel for kind of how these diagrams represent biological reality can be pretty useful. A little bit of background in biophysics. Again, a few points that you may want to look into. Probably the most important thing on this list is having a base understanding of what diffusion is. So we'll have a couple of modules on diffusion in CompuCell, but understanding the principle of diffusion in advance of the workshop sometimes can be useful because it's used a lot in CompuCell simulations. So at the bottom right here, we have the diffusion equation. There's some images of kind of the basic principles of diffusion. Definitely recommend at least sort of taking a Wikipedia level scan of the concept of diffusion if you've never seen it before. For the background in mathematics, a couple of things here again. to get you thinking, but the most important is probably a basic familiar with first order ordinary differential equations and rate laws. The rate laws will be especially important when we get to the network modeling portion of the workshop. But the rest of the models are run using first order ODEs. So understanding what an ordinary differential equation is can be pretty useful. Again, you don't need to be an expert in ordinary differential equations, but just knowing what it is and kind of the general structure of a first order ODE can be very helpful, especially once you start getting into troubleshooting and also thinking about how to set up your system to represent the model that you want. So we have some resources here for places to fill in some of this background if you are interested in spending some time over the next week or two looking into this. Again, these links are all available in the slide deck, which is available in the student materials folder. I know we've had a couple people join since I shared those, so I will put that in the chat again, actually. The link to the student materials folder. And then I'll just pop the Slack link in here as well. So if you haven't joined the Slack yet, here's the link again for the Slack workspace. But we do have some resources here if you're interested in learning a little bit more about each of these areas. We have a couple of papers here on the modeling process more generally that we think are useful to read in advance of the workshop. We have some resources for learning Python. In the past, we've recommended this Python Village 6-exercise tutorial from Rosalind. If you can do exercises 1 through 5 in the Python Village, we think you're in pretty good shape for both participating in the workshop and in the hackathon. And then we have some other resources here for where to fill in the rest of your background if you would like to do some casual reading before the workshop. Again, none of this is strictly necessary. We will teach you everything you need to know to use CompuCell during the workshop. Again, like we said, if you do a little bit of reading ahead of time, sometimes it helps with keeping those concepts from all being super unfamiliar when you actually get into the workshop. All right, so other resources for the class. This is actual like course materials stuff. So again, everything can be found through our website, CompuCell3D.org. The course website here, Workshop 24. We will post announcements to the website as well as updated links to meetings. We will be putting a schedule of the actual course topics up later today on the website. That will also go out by email with the announcements email later tonight. So keep an eye out there. You can download CompuCell from CompuCell3D.org/sourcebin. We have some help manuals there as well. So we have the CompuCell reference manual, which talks about the basics of how to use CompuCell, simulation specification, cell properties, things like that. We also have other help files available. We have a Python scripting manual which is a list of how to do a number of common things in CompuCell using the Python side of things, as well as a quick start guide that will help you with setting up a couple of very basic simulations so you can kind of get some hands-on experience with building your first couple of CompuCell simulations. We'll do one of those today at the end of the session here. Again, we'll have our Slack channel and the course Google Drive where all of our student materials will be posted. There are a couple of quick reference guides as well. These were sent in your acceptance letter in a zipped file, but they're also available through the student materials folder at the link above. We find these reference manuals to be very useful. We have one on Python, basic functions in CompuCell, as well as Tellurium. Once we get to the network modeling portion of the workshop, the Tellurium manual will be especially useful. We find it useful to have these either printed out or saved somewhere on your desktop where they're easy to find if you do need to reference them quickly during an activity. Again, we'll have our Slack channel. You can always ask questions through Zoom's chat. However, Zoom chat is not permanent, as we all know, lost when the session ends. The Slack is a more permanent-ish space for asking questions. We say permanent-ish because the Slack will only be available for about 60 days after the workshop. So you'll be able to use the Slack to continue discussing things for a little while after the workshop. But once that kind of 60-day period is up, you will have to make sure that you've saved off anything that you want to keep long term. And we'll make sure to send announcements at least a week or two in advance before that workshop becomes inaccessible. That way you have a chance to grab anything that you needed off of there. We'll have different channels for different topics. People often use this for file sharing, especially during the hackathon. It's a good centralized place for that. You can create um, your own channels for your hackathon group. We'll talk about how to do that during the actual workshop. It's also a good place to ask questions if you need help about things that maybe aren't being directly discussed in the day's sessions, or if you're having technical help or technical issues that you need help with CompuCell. The Slack is a good place to do that. We'll have people monitoring that periodically throughout each day. It's also a good place to get help from your peers. Somebody may be having an issue, similar issue that they've already solved, and they may be able to get around to helping you before the instructors can check for the day. So sometimes it's an even faster way to get help with that in mind. We do have some extra readings available in the course drive if you'd like to do a little bit of reading before the workshop. We have a few papers in there that you can take a look at. Again, some on the basic modeling process, some on the history of CompuCell. We have a couple of published papers that we think are especially good examples of CompuCell simulations. So if you'd like to take a look around some of those papers, those are available as well. And we may be putting additional papers in this folder as we move on through the workshop. If you have papers that you think are also especially useful resources, please feel free to email them to me, and we may consider putting those in the readings folder. All right, any questions about resources or anything before we move on to some quick overview of the hackathon and then our CompuCell demo? Okay, so again, if questions come up, feel free to leave them in the chat. I'll be checking that periodically. So some quick details about the hackathon. So we will be hosting a model building hackathon after the workshop. This is a chance for people to form groups to work on larger projects for a couple of days. And the idea is to sort of build a functional prototype of your model by the end of that weekend. The way this works, so the workshop is taking place between July 28th and August 4th. We'll have a short break in between the workshop and the hackathon this year. And then we'll resume the hackathon on August 9th. So there'll be about 5 days between the workshop and the hackathon. And then that'll go through Sunday, August 11th. That Friday will probably start in the mid to late afternoon. The main purpose of the Friday will be to finalize organizing project teams, figure out what everybody's working on, try to get everybody set up and structured and ready to go. And then on Saturday, we'll start with the actual model building portion of the hackathon. We will be organizing several project pitch meetings. There will be an opportunity at the tail end of the hackathon or at the tail end of the workshop during the extra sessions before lunch to do some lightning talks of 3 to 5 minutes each for you to pitch your idea, start doing some teaming, tell people what you're working on and see if anybody's interested. We will also host a couple of teaming meetings during that week between the workshop and the hackathon to help people find groups before the hackathon starts. And we will send those dates out once we have determined exactly when those will be. We'll provide you a template if you would like to do a lightning talk for it's like I think a 5-slide template that we've sent around in the past that people use to structure their talk. And then we can help select, we say here, select a preliminary list of feasible projects. We're not going to say what you can and can't work on. What we mean here is that we'll help you. If you have a couple of different ideas, we can help you pick the idea that we think is most accomplishable during the hackathon. Or if you have an idea that is larger than 2 days of work, we can help narrow that down into something that can be prototyped over the course of the weekend. So if you have an idea, but you're not sure how to sort of conceptualize it down to something that can be accomplished within 2 days, please feel free to contact us and we're happy to help you with the project scoping piece of that. If you don't have a project idea, but you would like to participate in the hackathon, we're happy to help you find a group. Again, some of this will happen naturally over the course of the workshop, but if you're interested in a couple of different things, you can send us, which top 3 projects you'd like to work on, and we can help you find a group depending on which group needs what. So if you're looking to join a group, but you're not sure how to go about doing that, please feel free to let us know and we can help you with that process as well. During the actual hackathon, we'll have group check-ins in the morning and at noon to kind of get a feel for where everybody is in the process, what kind of feedback they need, what kind of help they might need from specific instructors. We'll have the instructors in the room kind of circulating, working with different groups to help you build your models. And then in the evening on Saturday, we'll have an evening report so we can get you feedback for the next morning. And then on the evening on Sunday, we'll have kind of like a final presentations, informal presentations thing where you can show off, the current state of your model prototype. Post-hackathon support is available with the CompuCell team after the hackathon. So if this is something that you'd like to turn into a publishable paper, or if it's something that you're starting for thesis work, or even if it's just something that you would like to continue pursuing as a personal interest or hobby, we're happy to continue working with you after the hackathon on sort of expanding on your model and turning it into something, publication worthy. If you're not sure what to do, but you think that you would like to work on a project of your own, we've found that reproducing existing models using CompuCell is a great project if you don't already have one in mind. On the next slide, I believe we have a list of papers that we think are good candidates for turning into CompuCell simulations. So that can help take some of the sort of biological heavy lifting out of the process if you're not as familiar with, if you don't have a biological problem that you're already interested in, this is kind of a good way to get started. Replicating an existing model is also a good way to do it. There's a couple of software packages similar to CompuCell. There's one called Morpheus, a couple others that use the same underlying methodology but are different enough that it's worth the time to convert into CompuCell. So that's also a good way to do it if you're interested in just trying to translate an existing simulation into something that will run in CompuCell. That's also a good idea. For getting some hands-on experience with working with CompuCell and the modeling process more generally. So again, we'll have the hackathon teaming document sent out tomorrow along with the survey of your current interests in participating in the hackathon. And then we have that list of possible reproduction project ideas here in the slide deck. Couple things about hackathon expectations. If you are interested in leading a project group, we do expect you to have some idea of what you want to model and why. So coming up with a couple of questions that you'd like to answer with your model is a good way to kind of target your hackathon experience. If you would like to participate in a group, if you don't have an idea of your own, some sort of shareable description of yourself and your skill set and background is usually very useful. It helps us team people with the right group. Again, it's okay if you're just a total beginner. That's perfectly fine. Even beginners have skills to offer to a group. But just some kind of list of here's the things that I think are particularly relevant to the hackathon group work environment is useful for us. And the teaming document that we'll send out tomorrow will have some space for you to do that, and we'll tell you how to use that when we send it out. Expectations you can have of us is that for that weekend, we will be available for questions and assistance throughout the hackathon. We can't necessarily promise 24/7 availability, but we try to be available as close to 24/7 as we can for those 2 work days. Because we know that particularly with groups working within different time zones, there's going to be some pretty weird hours that people are up working. So we do try to have people available in shifts for that. Any questions about the hackathon before we move on to the CompuCell tutorial? So just for your reference in terms of time here, this tutorial should take somewhere between 15 and 20 minutes to walk through. So we'll probably go over until about 1:10 to get through the whole tutorial. The slides are available if you'd like to walk through it on your own. But just to give people a heads up, if you had somewhere you needed to be at 1, we'll probably only make it through the first half of this tutorial before the official end of the session. And then I'll stay over to finish the tutorial for those who are interested in watching the whole thing. Okay, no questions about the hackathon so far. Again, if you think of something, feel free to drop it in the chat. And in the meantime, we will go ahead and get started on building our first CompuCell simulation. You take a drink of water real quick. Okay, so like I said at the beginning of the session today, if you don't have CompuCell installed currently, don't worry. You can just kind of watch along and then try it yourself later once you do get CompuCell installed. If you do have CompuCell installed currently, feel free to go ahead and open it up now and follow along with this. So first things first, CompuCell consists of two main tools, CompuCell Player and Twedit, which is a code editor. So Player is where simulations are actually run, where your output is shown, and where you can control things and modify parameters while the simulation is running. Twedit is where you will be doing all of your actual coding of the simulation, where the code files are modified. So there's two separate pieces, and they start separately in the software. It's important to know before we start. So again, installing running CompuCell3D. Not going to go into huge detail about this today. Again, it is important that you do have a working installation of CompuCell before the workshop starts. And if you're having trouble, please let us know as soon as possible, and we can set up a time to meet with you to get it fixed. Again, you can download from our source bin page the binaries. So for Windows, there will be a single installer. For Mac, there's currently two installers. If you have a newer Mac with an M chip, M1 through M3 chip, you need the M1 installer. If you have an older Mac that still had an Intel chip, you need the x86 installer. So there's two installers for Mac on the website. So please do keep that in mind. If you try to install the M1 installer on a x86 chip, it will not complete properly. All right, so for a first experience writing and running a CompuCell model, we're going to use Twedit today to create a cell growth simulation. This is not in NanoHub. This is in your local CompuCell installation. We do have some example simulations on NanoHub, but for this, we'll just be using our local install today. So I'm actually going to exit out. I'm going to share my whole screen here so you can see. I'm going to go ahead and get CompuCell started. So when you first launch CompuCell, you should see a terminal window up here. This terminal will stay open the whole time you're using CompuCell. It's important not to close it. Closing the terminal will close everything you're doing in CompuCell. I've learned that the hard way a few times, frantically exiting out of things to save RAM. So do leave this window open. It's also a good way to check and see what CompuCell is doing. What CompuCell is doing in the background will be displayed on the terminal. So if you've clicked on a command and it seems like it's hung up, you can check the terminal to see what's actually happening in the background. So when you first start CompuCell, you should see the player window. Everybody see my player window okay here? Make sure the screen share is working. Somebody could just give me a thumbs up. Yeah, okay, great. Thank you. All right, so this is our player window. In the middle here in this big gray box is where your simulation output will be displayed. Currently, we don't have anything loaded in. We technically do. It opens up typically your last simulation. When you first open CompuCell, though, nothing will be running. We have a question here, how to open CompuCell. Are you in Windows or Mac, Ketchum? Mac, okay. So let's I can show you how to open it during the Q&A session at the end. For now, just watch along because we're running a little bit short on time. And depending on how CompuCell installed to your Mac, it's not always super straightforward. So for now, we're in our player window. And the first thing we're going to do, we're going to open an example simulation, and then I'll walk you through building a slightly different simulation. So to open a CompuCell simulation, you just go to File, Open Simulation File. CompuCell will come packaged with a number of demos. Should be in your CompuCell3D folder. You've got a folder called Demos. There's a number of different things you can look at. Each of these folders is kind of a different topic in CompuCell. We're going to look today in the book chapter demos at the angiogenesis simulation. So it's in CompuCell3D, demos, book chapter demos, angiogenesis. You'll double click on the folder, and then you'll just open the angiogenesis.cc3d file. The way that this works is that the simulation code is actually stored in the simulation folder, but you'll just open the CompuCell file, and it will open everything it needs for you. So you click open. And when you click open, nothing will seem to happen, but you'll notice at the top bar, it did change to the path for this angiogenesis CC3D file. So we just need to hit play or run to actually run the simulation. Sometimes the first time you start CompuCell, it takes a minute to run. This time it did not. So here what we're looking at is a three-dimensional bundle of cells that are forming blood vessel vasculature over time. So if you're following along, you should see something along these lines. Sometimes it'll start in 2D format. So if you're seeing something like this, what that means is you're just looking at the XY plane. You can go up in your graphics window to this drop down and click on 3D. One thing to note is when you change viewing types, it will pause the simulation. So you'll have to come back up and press run again to resume. A couple notes about stopping a simulation. So there's two ways to stop a simulation. You can actually stop. So if you click the stop button, the simulation actually closes and quits. Now, when you hit run again, you'll notice it starts again from the beginning. It reloads the whole thing from the initial parameters. Sometimes a more useful way to stop a simulation is actually just to pause it. And if we click pause up here in the upper left, that'll actually just freeze the simulation at its current Monte Carlo step, which allows you to go in. If you wanted to change parameters in real time, you can do that. And then when you hit play, again, it will resume from its previous step. So just something important to keep in mind there. Pausing is if you just want to stop, but you want to resume. But if you click stop, you'll actually start from the beginning parameters again. So that's how, if you want to change the color of cells, you can do so over here. Make these a little bit brighter. And then here we can steer the simulation. So if we wanted to change our target volume, like how big the cells are trying to be, let's bump that up to 100. And you can see the cells actually change in real time. If we wanted to bump that down to 50, see them shrink down. There's a number of different things you can steer once you've started the simulation. Okay, so again, this is also a good way to test if your CompuCell installed properly. If you can open a demo and run it, you can now use this angiogenesis demo to make sure it's doing what it's supposed to be doing. So that's how to open, and there's a bunch of different demos if you wanted to look through the various demos that come packaged with CompuCell. There's a number of different things you can take a look at. But today we want to go ahead and walk through actually building our own simulation. So to do that, we need to start Twedit. Twedit is not started by default. So there's two ways to do that. You can either go to File, Start Twedit, or from the menu bar up here, this icon with the paper and pencil will also open Twedit. And again, sometimes it's quick for me today because it's already in RAM. Sometimes if it takes, on your first time opening CompuCell, it may take a minute to open Twedit. So this is Twedit. This is our code editor for actually building and modifying CompuCell simulations. We have our editor window here in the middle, project manager on the left, and then our toolbar at the top, like most um, IDEs that you would work with. For today, we're going to go to CC3D project. You can also, if you wanted to open the demos that are bundled with CompuCell in Twedit and look at the code, you can do that here just by following the same process you would in Player, but in Twedit. Today, though, we're going to make our own simulation. So we're going to go to New CompuCell3D project. And what that does is it opens up our simulation creation wizard. This wizard will walk you through a number of different menus where you can pick some initial configuration parameters, and then it will actually generate all of the code files you need to run a CompuCell simulation. And you can just modify from there, so you're not having to code from scratch. So first things first, we need to give our simulation a name and pick where to save it. Some notes on that. You may need to make sure that when you're saving to a directory, that it is a directory where you have full write access. If you do not have write access to the folder where you're trying to save, CompuCell can't create the temp files it needs to run the simulation, and it will often crash without useful information. So make sure you're saving somewhere like your desktop, anywhere in your user folder where you have full read-write access. So I've already got mine set up, and then we need to give it a name. Some details on naming. I think we have a slide on this in the slide deck. Your simulation cannot start with a number. The simulation name can include numbers, it just can't begin with one. So we're going to call this growth sim. CompuCell names also cannot include spaces, but you can use underscores. So we're going to call this V2 because this is the second time I've done this today. It also tends to have problems with special characters, so things like hyphens, periods, exclamation points, parentheses, things like that. Try to avoid those in your CompuCell names. Just try to stick to alphanumeric characters, no spaces, and don't begin the name with a number. Those are the three big rules. We're going to go ahead and click next. We want Python plus XML. Next window is our general simulation properties. So this determines the properties of the simulation lattice. Again, we'll talk more during the workshop about what each of these functions do. For now, we're going to leave this all as default though. So we're going to leave this as a lattice of 256 pixels in the X direction, 256 pixels in the Y direction, and then 1 pixel in the Z direction. So this is a 2D simulation. We're going to leave all of the rest of this the same. And then we're going to start with our initial cell layout as rectangular slab. Click next. Next menu is the cell type specification. So this is where we determine which cells we want in our simulation. So it will always start with cell type of medium. The medium is what the rest of the cells sit in. So you will always need a medium cell. And then we're going to add two different types of cells. We're going to call the first one cell 1. Click add. You'll notice when you add, it gets added to the table up here. And we're going to call our second one cell 2, and you can also just press enter after you type it to add it to the table. So once the cell is populated in this table, it means it's been officially added to the simulation. We don't want any of our cells frozen. We do want them to move around. So we're going to leave freeze unchecked. So go ahead and click next. Chemical fields we can specify on the next page. We're not going to use one in today's example, so if we just click next without filling anything out, it will leave it empty for us. The next page is kind of the main page of the wizard. This is where we pick our cell properties and behaviors plugins. So this determines what different types of features your cells have. Starting with our cellular behaviors, for today, we're going to pick contact because we want the cells to stick to each other. We're going to pick growth. And you'll notice when I click growth, some of the constraints and forces options disappear because they're not all compatible with the growth plugin. So good practice is to pick your cellular behaviors first and see if anything gets narrowed down over here. We want our cells to grow, and then we want our cells to divide. So we're going to pick mitosis as well. And we also need a volume constraint because we want to control how big our cells get. And then those 4 will actually be the 4 we need for our cell growth and division example. So we'll leave the rest of it as it was. We'll click next. You'll get a configuration completed window. You still have the opportunity here to go back and change anything if you need to, but when you click finish, it will actually create the project and save it to your hard drive. So we'll go ahead and click finish. And if you're following along, congratulations, we've just built our first CompuCell simulation. You'll notice it populated the CD or CC3D project menu with our project. You can drop down to see which files were built. And if we want to view these in Twedit, we just double click on the project file and it will put the files in our editor for us. So there's three primary files for each CompuCell simulation. There's one XML file that specifies the simulation at startup, one Python file for actually telling the computer to run the simulation, and then a stepables file also in Python, which is where parameters that are controlled over time as the simulation runs are maintained. So just moving from left to right, we'll start with a quick look at the XML, and you'll notice here if you take a look through the code that the values that we specified in the wizard have just been added to this XML file automatically. So we have our dimensions of our lattice here, number of Monte Carlo steps, neighbor order. We have the cell types we specified here. So we've got medium, cell 1, and cell 2. We have our contact plugin that we chose here, specifying contact energies between each cell type. We're going to leave those all at 10 for now. And then we have our uniform initializer, specifying a rectangular blob in the middle. Again, we'll go over during the workshop what each of these things is in more detail. But for now, this is just showing you that the things that we were specifying in Wizard have been dropped into the code automatically for us. So our simulation Python file, again, this is just telling the computer to run the simulation and how to run it. You will almost never need to edit this file, at least with what we're doing during the workshop. What you will need to edit, though, is the stepables file. So this is, again, where all of our Python sits. And these are functions that control things at initialization and startup of a simulation. In between each specified number of steps in the simulation, it will update certain properties of the cells. And then what happens at the end of a simulation as well. So here you'll notice we have our mitosis stepable that was added from Wizard. If cell volume is greater than 50, the cells divide. Then we have our cell growth stepable as well that adds volume to cells after each certain number of Monte Carlo steps and our volume constraint here. So we don't need to modify anything today to make this run. To run it in Player, we just hover, right click, and then open in Player. And then this should pop up new player window. Here we go. And the simulation should automatically start running. You'll see the cells spreading out. And if you kind of focus on an individual region of cells, you'll see that as they grow, once they reach a certain size, they split into two cells. It's kind of where you get this sort of undulating effect. Where cells divide and it creates more room for other cells to grow, which then divide. So I'll go ahead and stop this and start it over. And again, if you're interested in trying to steer some of these properties over time, you can. So if we wanted to change contact energy between different cell types, we could. I won't mess around with that today because we're over time. But if you wanted to look at that on your own, you can. So that's what we have for our example today. I'll hang around and answer questions after if people are interested. I've got one more slide to look at. Again, if you would like to walk through this exercise on your own after the fact, we have step-by-step instructions on how to do exactly what I just did in the slide deck for today. In addition, we also have some slides on a little bit more detail on the actual code specifications if you wanted to take a look at what each portion of the code is doing in more detail. There's some slides there on how the code corresponds to what you're looking at in the output. So last thing for today, things to do to prepare for the course. So again, make sure that you've installed CompuCell and that you can launch it and that it runs properly. Double check that you can load in a demo. A couple of good ones to try, again, are the angiogenesis demo that I demonstrated a minute ago. Another good one is this delta notch demo. Delta notch is good because it will also make sure that your tellurium and network modeling functions are working properly. So if you can launch both of these without errors, you're in good shape for the workshop. Um, Again, do any reviewing of background reading that you would like. If there are things that you don't feel super comfortable with, again, you don't need to be an expert in any of these ideas, but seeing them ahead of time can sometimes help with making learning the content during the workshop a little bit smoother. Again, we will have a quick survey sent out tomorrow for you to fill out about your hackathon interests, as well as the teaming document. So keep an eye on your emails for that. And I think with that, we can just move into Q&A session. So if you don't have any specific questions today, thank you for coming. That's all we have to talk about. Again, just keep an eye out on your email for upcoming announcements. And we look forward to working with you at the end of this month. But I will stick around for other questions in the meantime. So I know we have one. Shanila, am I pronouncing that correctly?
Yes.
Okay. Repeat the last step. So which step did you need?
When we created this simulation file, and now how we run it again in this Compu.
Right. So you have this, right? You have the code.
Yes.
Yeah, if you could just go to growthsimv2.cc3d in the project manager here,
Mm-hmm.
and then you right click and then open in player. And that will
Yeah.
send the code to player from Twedit, and it should start running automatically.
Okay, thank you so much.
Mhm. So, Katrin, it looks like Karthik has a YouTube tutorial that he can send you. And we will
Uh,
be,
I I installed the package actually, and the website says it's installed in my folder, in my personal folder, in the Katrin folder on my Mac. And this is right, and everything worked perfect. And now I have a bunch of yeah files in there. One is CompuCell3D command, which appears to be a script, and then demos, install log, miniconda 3, param scan command, run script command, and widget command.
Mhm.
But no, nothing where I can just start the application.
Right. Does running the CompuCell command start it?
Um, loading user command exit, loading CompuCell3D player using app. Yeah, something happens.
Okay. Would you be comfortable sharing your screen for just a minute?
Hmm. I cannot. Okay, now I could.
Sorry, I always forget I have to stop mine.
So this is the window that appeared
Mhm.
when I clicked on this CompuCell command thing.
Okay. So I am not a Mac user. We do have some people in the group who might be better suited to help with this.
I hear something
Oh,
happens in this window.
there you go. Okay,
Ah,
yeah. Sometimes it just
okay.
takes a minute to start, especially if it's the first time you've started like that day since you've turned your computer off. Um,
Yeah.
Sometimes you
Okay.
just have to
So,
wait a minute.
yeah. Thanks a lot. I will give my very best to try it.
Yeah,
Thank
yeah, no
you.
problem. And if you have any issues with the example, just let me know and we can touch base on that.
Yeah, thank you.
Mhm. Even, are you on Mac or PC? Yeah, so Stephen, what I might recommend is just uninstalling and trying to reinstall it. Um, if you can. If you want to send me an email, I can try to forward this to the people who may be able to help. If you can have the full error command and everything, that would be useful. Yeah. Yeah. So please, if you can't get it installed, do email us. I think right now we're working on trying to schedule at least one more installation help session for people. Because I know that now that the workshop's coming up, people are, trying to install it and facing new issues and things like that. So we'll send out an email about that, about when that will be held as well. We usually try to schedule a large window so people can make it. Yeah, Karthik, if you're still here, would you mind sending that? Oh, okay. Thank you, Stephen. All right. Any other questions?
I had a question.
Mhm.
Actually, I had a couple of them. So I was wondering, so all the demos that I've watched so far on the CompuCell3D source files, they're all kind of like compartmental models or agent-based models. I was wondering if it can do anything like finite elements or hyperelasticity, where you can model the tissue mechanics of a nonlinear stress-strain relation using a strain energy density function.
Um, hmm, let me think. So I know you can measure the stress and strain within cells with CompuCell. I'm not sure, depending on how close to finite element analysis you want it to be. Um, I'm not sure about that, but I know that you can definitely measure what's going on mechanically with the cells. Um,
Okay.
it just requires a little bit of clever application of a couple different plugins. Um,
Okay. And also, I have another question. So, do you know if CompuCell 3D does any kind of like, or has any kind of like plugins that can do image analysis? For example, if I have a sequence of images from slides or experiments, right? And I want to track the motion of a particular cell through all those images.
mhm.
Are there any plugins in CompuCell 3D that can do that?
Um, that's a good question. I don't exactly know the answer to that. I don't think off the top of my head, I don't think there's anything built into CompuCell that could just look at pictures. Obviously, if we wanted to build a simulation that mimicked those pictures, we could do that. But as far as like just taking in images and doing analysis on that, I'm not 100% sure. But if you want to send me an email with both of those questions, I can definitely put you in touch with people who may be able to answer those
Yeah,
more
I'll send you.
completely.
I'll send you. Thank you so much.
Because I know we have a couple of people doing work using machine learning to analyze output information from CompuCell. So it's like you have a bunch of different possible simulation states and using machine learning to kind of identify the ones that are biologically useful. So CompuCell may not do it, but they may be able to give you some information on packages that they've been using for doing that kind of image analysis.
Also, I'll send you an email.
Great.
Thank you so much.
Yeah, no problem. Other questions? All right. Well, we are a bit over time now. So I think if there aren't any other questions currently in the room, I will go ahead and call it here. So thank you everybody for coming. If anything does come up, if you think of more questions along the way, please just feel free to email me at any point, and we will stay in touch. So, all right. I look forward to seeing everybody in just over a week, and we will talk to you soon.